A very good day to everyone. We are from Group Helix Hunters. Let's have a look into Introduction of Genetics. What is genetics? Genetics is the science that deals with the study of genes, heredity, and the variation of organisms and tries to explain what they are and how they work. It explains how traits, like eye color, height, or even certain diseases, are inherited from parents. One key concept in genetics is heredity, which is the process of passing traits from parents to offspring. For example, you might inherit your father's nose shape or your mother's dimples. Another important concept is variation, which explains the differences and similarities among organisms. This is why siblings can look alike but still have unique features that make them different. What are genes? A gene is a hereditary unit consisting of DNA that occupies a spot on a chromosome and determines a characteristic in an organism. The cell contains chromosomes, and each chromosome contains thousands of genes, which is 1,000 bases long. DNA is organized into specific units called genes. The sequence of bases in each gene contains instructions for making a single protein. Each protein serves a particular function in the body. For example, enzymes help us digest food by breaking down complex molecules. Structural elements give our cells shape, and signaling molecules help the cells communicate with each other. Coordinating processes like growth and repair. The DNA molecule. It composed of two polymers of nucleotides oriented in antiparallel form. The molecule resembles a spiral staircase of complementary base pairs which include a sugar, phosphate group, and nitrogenous bases. This is the nucleotide structure of DNA and RNA. Both the DNA and RNA contain three components: phosphate group, nitrogenous base, and sugar molecule. DNA contains deoxyribose sugar while RNA carries the ribose sugar. Nitrogenous bases in DNA are adenine, guanine, cytosine, and thymine, while RNA contains uracil instead of thymine. Double helix structure of DNA was proposed by Watson and Crick. Two strands run in opposite directions, known as antiparallel backbones. The nitrogenous bases pair correctly adenine with thymine and cytosine with guanine, forming the stable structure that stores genetic information. A chromosome is a strand of DNA that is encoded with genes. It constitutes an entire DNA molecule and protein. Humans contain 46 such molecules, 23 pairs of chromosomes. This is the human karyotype, consisting of 46 chromosomes, 44 somatic chromosomes, and 2 sex chromosomes. Females have 2 X chromosomes XX, while males have 1 X and 1 Y chromosome, XY. Genes constitute distinct regions on the chromosome. Each gene codes for a protein product. Differences in proteins brings about differences between individuals and species. This chart of rules state that in DNA, the amount of adenine equals the amount of thymine, and the amount of cytosine equals the amount of guanine. This 1-to-1 ratio ensures base pairing. These rules are crucial for maintaining the structure and integrity of the DNA double helix. The history of DNA discovery is filled with groundbreaking moments. In 1928, Frederick Griffith discovered that a factor in diseased bacteria can transform harmless bacteria into deadly bacteria. Rosalind Franklin captured an X-ray photo of DNA in 1952. Watson and Crick described the DNA molecule from Franklin's X-ray in the year of 1953. The conservative model of DNA replication states that the original DNA double helix is the template for a new double helix. This means that each round of cell division produces 1 new DNA double helix and 1 original DNA double helix. DNA helicase is involved in DNA replication, and it breaks hydrogen bonds between DNA strands. DNA polymerase joins free nucleotides into a new strand of DNA, while DNA ligase joins DNA segments on discontinuous strand. What is recombinant DNA? It is DNA that has been cut from one strand of DNA and then inserted into the gap of another piece of DNA that has been broken. The host DNA is often a bacterial cell such as E. coli. The purpose of splicing the gene into the host DNA is to produce many copies of it. As bacteria reproduce in a very short time, it is possible to make millions of copies of the gene fairly quickly. How do we make it? First, insulin is cut from the DNA using a restriction enzyme. A plasmid is removed from the bacterial cell and is cut open using the same restriction enzyme. Then, the cut-out human gene is then mixed with the bacterial plasmids in a test tube. The cut ends of the plasmid and the end of the human gene match and are often called sticky ends. The enzyme DNA ligase is used to stick the ends together, forming recombinant plasmid. After recombinant plasmid is formed, combine them in a test tube with CaCl2. The high concentration of calcium ions makes the membranes of the bacteria more porous. This then allows the plasmids to move into the bacterial cells. In the selection of altered cells, an antibiotic resistance gene is used to identify recombinant bacterial cells. Only recombinant plasmids containing genes and antibiotic resistance genes will grow. The desired genes can then be selected for further use. DNA has key properties that make it useful. The complementary strands of DNA can be separated and re-associated by heating and cooling. One strand of DNA specifies the sequence of the other strand, ensuring accurate replication and genetic information transfer. Polymerase chain reaction (PCR) is a technique widely used to make many copies of a specific DNA segment from a tiny sample. PCR process consists of three main steps. First, denaturation occurs, where the DNA is heated to separate the double-stranded DNA into two single strands. Next is the annealing step. Primers bind to their complementary sequences at the target region of the DNA. Finally, during elongation, DNA polymerase extends the primers and synthesizes new DNA strands, creating copies of the target sequence. Genetics is important in real-world applications, as it is used in production of insulin by amplifying the insulin gene for large-scale production. PCR also helps create recombinant vaccines by producing viral genes for immunization. In agriculture, PCR is used to make genetically modified crops with insect resistance. Additionally, genetics is crucial in forensic crime scene analysis for DNA profiling, helping to identify individuals based on DNA found at crime scenes. Thank you for your attention. Hope this video sparked your curiosity about genetics.